Good afternoon, everyone, and welcome to this week's NITheCS Micro-school. My name is Dean Brand. I'm a second-year PhD student at Stellenbosch University working on quantum computing with Professor Francesco Petruccioni, who's the director of NITheCS. And this week's speaker is Dr. Caroline Ross, who is a RNA biologist specializing in the integration of computational and experimental approaches to explore the complexities of gene regulation. She's a lecturer at the Department of Molecular and Cell Biology at the University of Cape Town, specializing in bioinformatics. And I'll let her elaborate more on all of the details of that during today's talk. The topic this week is on LncLOOM, which is a graph-based framework for detecting ultra-conserved functional motifs in rapidly evolving sequences. It is a Python-based framework, which is developed by Caroline, and I will leave the details to her to get into. Yeah, if there are any questions, then please feel free to leave them in the chat. And yes, with that, I will leave it to you, Caroline. Take it away.
Thanks, Dean. Okay, hi, everybody. So yes, today I'm going to be talking to you about LncLOOM, which is a graph-based algorithm or framework that I developed during my postdoc. And I did this with Igor Litsky at the Weizmann Institute. Just 1 second, because the presentation is not changing. Okay, here we go. All right, so before I tell you about LncLOOM, I'm just going to spend some time telling you about a specific type of RNA molecule that we call long non-coding RNAs, because the LncLOOM algorithm was really developed as a comparative sequence analysis tool to analyze these specific sequences. Okay, so what are long non-coding RNAs or lncRNAs? Well, these are RNA molecules that do not encode proteins. So when we talk about RNA, if you are familiar with the COVID vaccine, for example, it's an mRNA vaccine. So when we talk about mRNA, we're talking about RNA molecules that are then translated into a functional protein. And if you look at the number of human genes that we actually have, only about 31% of our genes are actually transcribed into mRNA and then translated into protein. The rest of our genes are actually non-coding RNA. And the largest group of this is the lncRNA molecules. And so if you actually start to look at their sequences, or you look at them in terms of their RNA molecular structure, and you compare them to protein coding genes or mRNA molecules, they are similar in that they are polyadenylated, they are capped, but as I said before, they do not encode functional protein. And then another major difference between mRNA and lncRNA molecules is that their sequences of our lncRNA genes are really, really not conserved. So if you look at your typical protein coding sequences and you compare it, say, the same protein sequence across many species, they will often have high sequence similarity. And this allows us to align them. And from there, we can actually infer the functional domains. Often these will have similar structures. But when you look at an alignment of lncRNAs, the sequences appear almost random and absolutely not related. So the major question is, well, how do we then know, or how can we understand how these functions of these lncRNA molecules have been encoded in these random sequences? Okay, and so I'll just give you a bit of numbers here. We have now, over the past 10 years, research into the specific class of RNA molecules has been done, and there's about over 20,000 of them have been identified in the human genome, and many of them have been studied. The majority have not been studied, but from the ones that have been studied, we know that they have diverse functions, and they really are implicated in many different diseases, and they play diverse roles in regulating gene expression. Okay, and so really the open questions are, what fraction of these molecules are really actually functionally important? And this question stems from the fact that their sequences are not conserved in evolution. So if they were functional, we would expect that there would be some sequence conservation. But what we do know from selective studies is that even though the sequence is not conserved, often they can still perform similar functions. And this is really, we want to understand how this works. We want to understand their mechanisms. We want to understand how their functions are encoded in their sequences. And we also want to develop some sort of algorithm or computational tools to start classifying this diverse class of molecules, just like we are able to classify the thousands of protein coding genes in our genome. Okay, and so this is just giving you a bit more of a perspective of the computational problem that we addressed when we developed NITheCS. So if you look at, this is a multiple sequence alignment of HSP90, and we have 3 species that I've shown here. It's human, mouse, and Plasmodium, which is malaria. You can see that even between human and Plasmodium, the HSP90 sequence is highly conserved. And this is then, of course, translated into a very similar protein, which then folds. And this is actually the superimposition of their protein structures. And this is really conserved because they are performing a similar function across these species that are really, really, evolutionary distant. Now, this is an example of a multiple sequence alignment of the chaser lncRNA. And you can see we really, other than the 5' end here, we really don't pick up much sequence similarity. On the other hand, if you actually look at where the chaser lncRNA is transcribed in our genome, and this is true for many lncRNAs, you'll see that although there's no sequence conservation, they have what we call syntenic conservation, meaning that they are transcribed or produced in the same position in the genome. And in the case of chaser, you'll see that it's always transcribed. So for those of you that are looking at a plot like this for the first time, this is an annotation of our RNA molecule. This is a gene, this is chaser, and then this is CHAD2 or CHD2, which is a protein coding gene. And then this is our RNA-seq. So these peaks would correspond to reads or read count from RNA-seq analysis. And this basically shows us expression across different tissues. And you can see if you look at this relative to CHD2 across all the different species. So this is a protein coding gene, which is highly conserved across vertebrates, and it is highly conserved at the sequence level. You can see that chaser is always conserved, always transcribed directly upstream and in very close proximity to CHD2. And we do know that chaser actually plays a role in regulating the expression of CHD2. That's its main function. But if you actually then try and align, for example, your human sequence with zebrafish using BLAST, it cannot find any significant similarity. So that was really the challenge. We know for sure by looking at RNA-seq, we know that chaser is there, it's being transcribed in the right place. We know that it's across these species, it has the same function of regulating CHD2 expression, but yet the sequences are so different. And so really what inspired LncLOOM was that we needed a new and a much more sensitive approach to find some sort of subtle conservation signals, and that we could then actually use these to sort of identify ortholog sequences in model organisms, and also to produce some sort of testable hypotheses that we could then perturb in the lab to actually try and study the mechanisms of these molecules. And with that, we developed LncLOOM. And of course, the major question was, well, how can we actually detect similarity in these random sequences, but detect it to a level that is beyond what is expected to occur by random chance? And so the idea of this was based on the fact that even though their sequences might appear to be really random, within the sea of random organized nucleotides, there might actually be very, very, very short regions or short chimeras, we call these chimeras, or motifs, that are actually there, but they've kind of just been hidden in the sea of randomness. And so what we decided to look for was to look for a combination of short conserved motifs that appear in the same order. And so this would allow any distance of random nucleotides to be in between them, but as long as we actually get this conserved combination. And this is the whole point of LncLOOM, is that by using this approach to search for a combination where the order is preserved, we can then detect something that's more likely to occur and to give some functional representation and less likely to occur by random chance. So if you actually assume equal distribution of nucleotides across a sequence, a sigma, which is a motif of 6 nucleotides, we would expect it to occur once randomly around every 4,000 nucleotides. But if we're actually looking for a combination of sigmas where we actually want the exact order to be conserved, this is much more likely to be functional and less likely to occur by chance. So that was the thinking behind this. And how did we actually achieve this? Well, we used a graph-based approach, and I will talk more about this on the 25th of November. Okay, and so basically the sequences, we take each of our sequences. So for example, this would be the human sequence, okay? And we order our sequences, which we can call them different layers, where each species is a layer. So this would be our human sequence. This would be our mouse sequence. This would be maybe a possum, for example. And we break these up into all possible consecutive k-mers. And we then actually build a graph where we place edges between identical k-mers. So we connect identical k-mers. And then we basically assign a value of either a 1 or a 0 to each edge. A 1 meaning it's allowed, a 0 meaning it's not allowed. And we impose this condition that the edges cannot intersect, because if they intersect, it means that the order of the motifs has changed. And once we have actually built this graph and we impose these constraints, we can then solve where we want to maximize the sum of the edges and therefore solve for the longest non-intersecting paths in the graph. And so something input shown up here, we would then load this into the integer linear programming solver, and it would produce an output that looks similar to this. Okay, so in reality, it's a little bit more complicated. We're dealing with sequences that are thousands of nucleotides long, so we get thousands of k-mers, and we end up with this massive complex graph with many, many edges. And really, we want to go from something that looks like this to something that's a little bit more elegant, and this would be our LncLOOM solution, where we can see that we have, as we get closer, for example, across mammals, we will have a little bit more conservation, where when we look more deeply across more distantly related species, only a subset of these motifs would be conserved. But from this, we can definitely identify short functional elements that are deeply conserved across different species. And from there, we can make the hypothesis that these motifs are actually underlie the function or the mechanism of these lncRNA molecules. Okay, so this is really just a schematic and an overview of what we applied this to. So for the input, as I said before, you would have sequences from different species, and we actually arrange these, you order them, starting from your reference sequence, or the species you're most interested in. For us, it was generally human. And then we would order our sequences with increasing evolutionary distance. So we would have all the mammals and all the way down to fish, and zebrafish, for example, would be our last sequence. And we would then load this into LncLOOM, and we would, the output would be this combination of conserved motifs. And we then actually annotate these using eclipse data to try and infer whether these motifs correspond to binding sites of RNA binding proteins, or if they correspond to seed sites of microRNAs, for example. And we ran this on many different lncRNAs to try and identify short conserved motifs. And we also ran this on thousands of 3' UTR sequences, which are generally much less conserved compared to the protein coding regions. Okay, so I'll just give you a brief overview of where to find LncLOOM, how to run it, And I'll speak more about the algorithm on the 25th, and I will give some examples of results also on the 25th. Okay, so LncLOOM is implemented in Python 2 and Python 3, and it is available on GitHub. Here is the link. And so when you actually download this and you actually clone this GitHub repository, you get all the Python scripts that are required to run LncLOOM. This comes along with its own setup script, which will actually, you run this using pip, and it installs LncLOOM along with many of the dependencies that are needed. You also include in this download, you get the data files for annotation, specifically the microRNA annotation files. And then unfortunately, because the eClip data and the reference genome files are very large, we don't store them on GitHub. But there is a really nice documentation which will take you through installing this. So you can use your own eClip data and whatever reference genome you would want to use for your species of interest. And you just tell LncLOOM where to find it. And that would then be able to annotate based on your own data. And you also get the BLAT executable. And this is basically just to map those motifs back to positions in the genome and then to correspond it to the eClip data, for example. Okay, and so this is just taking you through this. First, you need to actually download the repository. You do this using git clone. Once you have this, you can then actually just simply install it using pip. So I've given examples. Both on GitHub will take you through installing for Python 2 or installing for Python 3. And once you've actually installed this, it should install the following dependencies: NetworkX, Pulp, NumPy, and a few others. If for whatever reason the pip installation fails, you can then install these individually, because you will need these Python packages to actually run LncLOOM. And again, all of these instructions have been given on the GitHub page. Okay, it also takes you through adding this to your path so that you don't have to call the executable every time. And yeah, and you will also need to install a couple extra dependencies that are not included in the setup path. This would be BLASTn and the morphed alignment. Okay, and so once you set up, you would then run LncLOOM, and it's very, very simple. It's a command line Linux-based interface. Okay, so firstly, I'll just talk about the actual input. The input is a FASTA file, and these are basically your mature RNA sequences. Okay, so these are your exons. And you can see I've ordered this starting with my query. The one that I'm most interested in is my human sequence, and I want to find motifs that are conserved from human to all these other relative species. And then I've tried to order my sequences by evolutionary distance. Okay. And you'll also notice, if you look closely, I've got X's here. These X's mark the exon boundaries. So, for example, we exclude all the introns, and we just mark this. You can put any number of X's, one X is enough. And you do not actually have to do this. If you just give them the full mature RNA sequence, it's enough. But if you do actually want to get some specificity on where your motifs are located and in which exons they are found, then you do need to specify your exon boundaries. So this is your very basic FASTA file input. Now, I do just want to mention, so LncLOOM, because it is a graph-based approach using integer linear programming, it becomes an NP hard problem. So we cannot do this for massive data sets. So it's most applicable if you have a specific lncRNA or a specific 3' UTR sequence that you're interested in. You maybe have 20 different sequences or 20 different species. It can handle up to about 10,000 nucleotides per sequence, but we can't do this on thousands and thousands of sequences. It just becomes too large, the graph becomes too big, and LncLOOM will actually just tell you that this is not feasible. So there does need to be some thought on how you actually construct your input. And then the very, very basic command to actually run this is just LncLOOM, and you use your faster option and you give it the path to your faster sequences. And this will run it with all the default options, which I think are pretty good most of the time. But there are several options that you can actually add to improve this. Okay, so one of the things you can do is say you actually now want to annotate your motifs. You want to find out if, for example, if any of them correspond to the binding sites of potential microRNAs. You would specify the target scan option. And this then takes all the seed sites from the target scan database, which is a database of your microRNAs across vertebrate species, and it will then actually annotate that. You can also do things like give it a project name, which means that all of your files would be stored in a particular folder for this particular data that you're running, and all your files would have the same prefix name, so it's really easy to kind of keep all your data together. If you want to annotate using eClip data to find out if any of these motifs actually match potential sites of RNA binding proteins, you can then annotate with eClip data, which this, again, you would have to download yourself. But as long as you tell LncLOOM where to find it, where you have saved your eClip data on your computer or your server, then this would be very straightforward. And then in terms of statistics, to actually try and get some sort of a statistical readout, what we actually do is we repeat this through many different iterations where we shuffle the sequences and generate random sequences, and you can specify this. So you can do however many iterations you want, 100 or 1,000. Obviously, it's just depending on how long you want to run this for. And then the statistical significance is actually empirically determined from these iterations. Okay, there are a few additional arguments that you can actually specify. So for example, similarity. So if you are dealing with very, very similar, if you have in your data set a few sequences that are very, very similar and they have high sequence similarity, you do not need LncLOOM for this. LncLOOM is really designed to pick up subtle sequence elements that are kind of hidden amongst sequences that do not have similarity. So it's advisable that you remove these highly similar sequences, because you're just going to have many, many, many edges and many, many identical k-mers connected, and your graph is going to get very, very large, and this will increase the computational complexity of the problem. So the default is to remove anything that is 100% similar, but you can change this if you see that LncLOOM is taking too long to run. And for sequences that are very similar, you can just run a blast. Okay, you can then also alter the length of the, or you can actually tell LncLOOM that you want to, for example, only find motifs that are conserved to a certain depth, for example. So if you have 10 sequences and you only want to look across mammals, and you have your first 5, for example, are mammalian, then you could specify this. You can also tell it which solver to use. So LncLOOM does use a Jarobi solver, which is a very, very fast, commercially available linear programming solver, and it is much, much faster. So I do recommend that you use this. But yeah, again, you would have to actually install it yourself, and you would have to have your own license to use it. You can get an academic license for free. And then you can actually just specify the start and the stop widths of the motifs. So the way LncLOOM works is it will first search for longer motifs. So the default is 15. So it will look for all k-mers that are conserved that have a length of 15. If it finds nothing, it then looks for motifs that are 14, 13, and so on and so on. It will go all the way down to a minimum of 6 bases. We don't recommend going beyond this, because as soon as you start getting to much shorter k-mers, you basically don't find them everywhere, and it's going to be much more difficult to sort of say that these are functionally significant. Okay, and then just in terms of the stats, so how is this actually determined? What we do, so we have 4 different statistical values. Okay, so you see I've got PI and EI, and PR and ER. Okay. So firstly, if I'm talking about R, then I'm talking about looking for these motifs in a set of random sequences, but where they have similar percentage identities across the layers. Okay, so these are random, but let's say the human sequence to the mouse sequence in our original data had 50% identity, then the first and the second layer in these random sequences would also have 50% identity. So we calculate the probability of finding an exact match to that motif in a set of random sequences where the identities are preserved. And we also calculate the probability of finding any combination of motifs, okay, any combination of the same number of motifs of the same length. So these can be motifs or k-mers that do not actually match our actual k-mer, but they are the same length, and they are found, for example, in the same number of combinations. So if we find in our sequences, for example, a set of a combination that contains 3 conserved motifs, then we would look to see if we can find, and each of these motifs, for example, of 7 nucleotides, and we would look and see if we can actually find another set of conserved, another conserved combination of any k-mers of 7 nucleotides. Okay, and then, so we do this firstly for a set of sequences where we preserve the similarity, and then our R values are very similar, except instead of preserving the similarity across the sequences, we preserve the dinucleotide composition. Okay. Right, and then in terms of the output, so we get 3 basic output files. The first is a bed file. This is a really nice visualization file. You can upload this to the genome browser, and this basically indicates the conservation of every motif. I will show you an example of this, and it's color-coded, which is really nice. You can visualize this alongside your RNA-seq data or your genome annotations. You get a folder of annotated HTML files, which allows you to visualize these motifs and actually navigate through all the output files. And then you also get a text file, which is basically all the information, all the motifs, the depth of each motif, and the respective annotations for each motif. And this just allows you, if you want to, as a bioinformatician, to actually then do some further analysis. You can then process this quite easily and do some string handling on it. Okay, so this is how the bed file output looks. So this is our results for the chaser lncRNA. And you can see I've zoomed in here to the last exon of chaser, which is where we found some really nice, deeply conserved motifs. And so the darker the brown color, the more conserved this region is. And this is about 6 bases. This is about 6 bases. This is about 6 bases. So it's just a really nice visualization output. Okay, and this is our HTML. So yeah, I'm actually looking at, this is my human sequence, and the motifs are colored in terms of their conservation. So blue, starting this bright blue, means that a motif is conserved in only 2 species, versus our dark red, it's conserved in 16 vertebrate species. And this particular example we ran on sequences from human, mouse, dog, all the way down to zebrafish. And if you actually then just hover on these motifs on the HTML file, you get this drop-down menu. It tells you your statistical values, if it was actually identified or predicted to have statistical significance. And it tells you the eclipse matches. This is the background. So for example, this particular RNA binding protein, yes, this motif does correspond to a peak, an eclipse peak for this particular protein, but you'll see that this protein was actually found to kind of bind 20% of the sequence anyways. So it's not really specific to this motif, where this one in capitals, so anything less than 10 would be shown in capitals. This is probably much more specific to the motif. And if you would also have, if you've run target scan, you would have your eclipse matches and your target scan results. And if you actually click on this, it would then take you to a much more detailed output. It tells you the motif, it tells you the fold enrichment, over the negative control. It tells you the cell lines, the strands, and everything, and also the background value. Okay, and then this is just a very similar result for target scan. So this tells you, for example, that this motif matches a seed site specific for this particular RNA. microRNA. This microRNA is conserved across mammalian species, and it tells you actually the extent of the match. And then the other thing that that LncLOOM does, it gives you what we call a module output. So this is basically just a visualization of where these motifs are located. So you'd see this is the start. So in the human sequence, you have this highly conserved motif here, starting at position 800, where in dog it starts at 340. So often your lncRNA sequences vary across species in terms of length, in terms of exon structure, number of exons. So they're very, very difficult to actually analyze. But what you can see here is that no matter where these actually start, you have the conserved motif, and then this is the distance. So you have 31 nucleotides that are not conserved. These are much more random, but you can see really the distance, the spacing between these two motifs is really, really conserved across mammalian species. And this is also the case between the second and the third motif here, with the exception of armadillo. And so we call this a module output, and this kind of just represents much more of a functional unit, how these motifs might be actually talking to each other to make some sort of a functional unit here. And this might be very useful. For example, if we just look at this, it might indicate that this is actually folding into some sort of secondary structure or RNA structure. So this is quite a nice way to visualize this. And then this is just an example of the text file output. And what we actually talk about here, this is a motif neighborhood, for example. It tells you the depth of this. So this was conserved across 16 species, but you can see the way in which it's being ordered. So the deeply conserved motif, which is very short, is only 6 bases, but you'll see that it's actually contained within longer k-mers that are conserved as the species actually become more closely related to each other. And so this just shows you the, so how these motifs are actually embedded in surrounding regions and how conserved these surrounding regions are across much more closely related species, and how it changes across as our evolutionary distance gets much bigger. And then this gives you your statistical values. It also tells you, for example, this is cut off, but it would tell you your target scan matches. And then it tells you for every motif, it also then gives you the flanking regions, left and right. So you could do some follow-up statistical analysis here, or sequence alignment, whatever question you would want to ask. Yes, and so with that, I'm happy to take questions, and I just want to mention Igor Olitsky. He was my postdoc advisor at the Weizmann Institute, and this was really his brainchild, and I was lucky enough to have the opportunity to actually code it. And so if you do want to use LncLOOM, it's very useful for lncRNA sequences, for UTRs, for looking at enhancer sequences, anything that is you expect to be less conserved than your protein coding genes. It's very useful, and you can email either of us if you have questions. Okay, thank you very much.
Thank you, Caroline. That was a very good and very thorough introduction to a very comprehensive Python package that I think will have a lot of usage in the field of bioinformatics and lncRNA analysis and really in computational biology. It's very, very impressive. And I see there are no questions waiting for us in the live meeting, but luckily you've put your contact information on this last slide, and I'm sure if people do have questions, then the GitHub page seems also very comprehensive and easy to interact with and post comments on. Yeah, I think that's very, very nice introduction. I'm very impressed, very excited to actually get started tinkering around with it myself as a nice introduction to the field of bioinformatics. But I'm looking forward to more of your presentations that you'll have within the NITheCS universe of presentations. Yeah.
Okay, thanks very much. And yeah, let me know how it goes if you're analyzing something interesting.
Yes, looking forward to more interactions. Thank you very much again for presenting today.
All right, cool.